This is Luca Comai with the lecture on QTL mapping. I'm going to use the data from East, who worked on Nicotiana using two lines, line A and line B, which differed in the flower length. Line A had 40 millimeter flowers, line B for 93 millimeter flowers. They're hybrid. The F1 has intermediate length, and so does the F2, but very different here is the fact that the F2 has a very broad distribution, while the parental and the F1 have a narrow distribution. The difference in variance correspond to the variance due to the genic effect, because the F2 have multiple different genotypes, while the F1 and the parentals have uniform genotypes. So all we're looking at is the environmental variance. East took these lines and selected through a number of generations for shorter corolla and longer corolla. And we can see the effect of that selection. So this is the table, the original table from his publication. Here is parent 1. We're looking at the phenotypic value shown in millimeters. So this is large corolla or long corolla, and this is short corolla. Here is parent 2. You can see the mean phenotypic values, about 92 and 40 for the 2. Here's the F1s, and then through selection for short and selection for long, we arrive to F5s that have very similar means of corolla length as the two parents. So here they are. So this tells us that there are genes controlling this trait. The appropriate allelic content of this gene can be selected. Now, where are these genes? So we are going to use this material to illustrate how QTL mapping can be done. What does QTL mean? QTL stands for Quantitative Trait Locus, and correspond to a locus on the chromosome that has an effect on the trait of interest, and the trait is a quantitative trait. Assuming that there are, for example, two loci controlling the corolla length, we can estimate the effect of each allele type. If we think, for example, that the alleles or the loci responsible for the trait of flower length are A and F, we can then attribute to the little a allele a certain phenotypic value, and the same to the little f, and then to the big A or big F. So let's call either big A or big F big Q, just to encompass both. And the phenotypic value would be the mean flower length divided by 4. If we have 4 copies of the uppercase allele, we would have therefore a corolla of 93. We divided that by 4. We have 23 millimeter contributed by the uppercase Q. Lowercase q with a corolla length of 40 would give us 10 millimeter per allele. We can calculate the expected output. So, for example, this genotype here, 1 big Q and 3 little q's, would give us 53 millimeter, and 3 large q as 1 small q would give us 79 millimeters. Just to clarify what I mean here, 1 big Q and 3 little q's is, for example, big A, little b, little b, and this could be big A, big B, little b. We have obtained F1 and F2, and now let's take those F2 and self them. Let's take just one plant from each selfed family and continue. And after a number of generations, we would find that we have reached a high degree of homozygosity. However, because in this line we have undergone recombination, each line generated through the process of inbreeding would be different from the others. So these are recombinant. They are inbred, and they are lines, so we can call them simply recombinant inbred lines. Sometimes this is abbreviated as RIL. To understand the contribution of the genome to the trait, we need to have an accurate method for genotyping. So let's assume that we have such method, and at the end I'll show one type of approach that would give us the type of genotyping we need. So this method is providing us a way to sample the genome on many different sites, let's say several hundred of sites, and at each site to be able to distinguish whether we have the blue DNA of one parent or the red DNA of the other parent. So the selfing of the F1 resulted in crossing over, so we may have perhaps two crossing over per chromosome. We would expect in the F2 to have this type of scrambled genomes. But notice that we don't yet have much homozygosity. We only expect half of the loci in the F2 to be homozygous, so the other half would be heterozygous. These are the type of plants that are then taken and self to achieve homozygosity. The homozygosity would increase with each generation, and we know that by the F8 our homozygosity would be equal to 1 minus 1/2 to the exponent of the number of inbreeding generation. So the first generation of inbreeding is from F1 to F2. So by the time we have an F8, we have inbred this individual, this group of plants 7 times, resulting in a 99.2% heterozygosity. So our plants would look like this. The genome of one plant is represented by the chromosome inside the rounded rectangle. And in this case, we see that we have portions that are derived from the blue parent and portions that are derived from the red parent. And of course, we have assumed that all the uppercase alleles come from the blue parent and all the lowercase alleles come from the red parent. We can actually, knowing what we have and assuming that the A locus and the F locus are responsible for the trait, we can calculate the contribution of these loci in each genotype and derive the expected distribution. So we are taking, for example, this plant here, which has the relevant genotype of having big A, big A, big F, big F. So this one has 4 of the uppercase units. We expect therefore the corolla length to be maximum, and if we take many of these plants and measure the corolla length, we would find that they would look distributed as indicated here with a mean somewhere around 93. If we take this other plant where we have genotype little a, little f, we know that the contribution of this allele would result in a corolla length which is much shorter. So here's the corolla length found the number of these individuals, again distributed around the mean, which is about 40. And we could derive similarly phenotypes for all of these. So in this case, we have done this with the knowledge of what the relevant loci are, but let's imagine that we don't know that. So what we would have, very good phenotypic value, very robust, because we've used multiple plants of the same genotype. That's one advantage of the recombinant inbred line. So we would get very good mean, and then correspondingly we would know what the genome is made. So you see, in the next step is take this knowledge and sample a locus across many lines, dividing the genotype into 2 types, one with the big case allele and the other one with the small case allele. So essentially we want to know for any one locus what is the effect of having the blue color or having the red color. This is called a scan. We look across the genome, and for any one locus we are evaluating the 2 types of genotype that we expect. So let's suppose that we look at a locus called M. For the locus called M, we might have about 150 plants that have big M, and these 150 plants would then be measured, and we would derive the mean for these 150 plants. So here's 150 plants that have big M. Of course, these plants would have all kinds of other different genotypes in the rest of the genome. So for the M locus, then we could look at the little m plants, and if we have developed about, let's say, 300 recombinant inbred line, we may also find that there are 150 plants with this genotype, again varying everywhere else in the genome. And for these 150 plants, we would measure the phenotype, see what the mean is. If the mean is the same for both genotypes, we would conclude that this locus has no effect on the trait. So let's imagine, for example, that now we are sampling locus A, so we have about 150 of our big A plants, and we know that the uppercase allele is the one that has the high value. When we look at the phenotype of all these 150 plants, we would find it distributed around the mean, which is of this value. Now, next to this comparison, we would look at the phenotype produced by having the little a allelic composition at the A locus, and say about 150 more of these plants, and we would get a phenotypic value that is shown that it has a smaller overall mean. So comparing these two mean, we would then apply a statistical test and determine whether it is significant or not. Let's suppose it's significant. Let's not worry about what the statistical test would be. Let's just say that in this case, we would have to conclude, therefore, that this particular locus has an effect on the trait. How would that look on a map? Well, if we imagine drawing a map of our QTL, a map of the candidate loci, that is the loci that have shown an association, the way we would derive that map would be to take the existing chromosomal map and then to plot on top of that the significance of the association. So let's say that this is my axis on which I would plot increasing significance of association. So as I plot this, it may look something like this. Perhaps I have a little peak here, it goes up and down, and then eventually as I approach A, we would get a peak that comes up and then comes down. I would need to establish a threshold of significance, some value above which I believe my statistical outcome, and that value may be, for example, here. It's not uncommon to use the logarithm of the odds, so this may correspond to an LOD value, let's say, of 7. You are familiar with what an LOD is, I don't need to explain it. Let's then look at the other chromosome. As we look at the other chromosome here, we would have again a significance, and then we would start by, let's say, from the top we have low significance, and then all of a sudden as we approach F, the significance would shoot up, cross the threshold, and we would know that in these regions the chromosome underlying it is having an effect on the trait. Having an effect on the trait means that there is one or more loci that are influencing that phenotypic trait, and so we would call this, let's say, QTL1 and this QTL2. What remains to be determined here, how do we get this very nice map? So the type of map that could be constructed is one based on some kind of molecular markers. For example, a commonly used marker is this one called variable number tandem repeat DNA marker. It's a PCR marker which would work as follows. Suppose I have a position on this chromosome corresponding perhaps to locus B, where I've identified the presence of a repeat. Now that repeat could be, for example, the sequence AG, and that repeat could be present, let's say, in a number of 10 copies. Next to that repeat, I would need unique DNA that can be used as the location for the primer complementarity. A primer would be would be designed that amplifies this sequence here from here to there. So here are the arrows representing my left and right primers. So this is the left primer, and this is the right primer. Amplification of this sequence would give me a PCR fragment that contains in its core the 10 repeats. I'm not going to draw all 10, something like that, each one of them being an AG repeat. These repeats are susceptible to change, not on a day-to-day basis, but over many generations. If we look at two lines, they are likely to have different number of repeats at that position. So if we, for example, were to look at the red line over here, we might find that in this site we have an AG repeat which is present, let's say, in 20 copies. And the flanking DNA is the same, so this can again be amplified, and I'm not going to draw it to scale, but here we would have the many 20 VNTR sites, or the repeat pattern of AG. Now, because this repeat is 20 bases larger due to the presence of 10 extra of these repeat, the overall fragment differ by 20 base pairs. So when I then take this system and load those PCR product, I would find that my blue band loaded here, my blue sample, my blue PCR a signal, a band that moves as far as here. We have a positive charge down here, so we have direction of movement as shown. And the red band loaded here would give me something which is about, what did I say, 20 base larger fragment. If, for example, we have an heterozygote where both fragments are being produced, I would then find this type of pattern where two bands are present. I'm drawing them in blue and red. They're not blue and red in actuality, they're just different in size. So identifying, and let me do away with the color, identifying this pattern here would tell me that I have the blue DNA. Identifying this type of band would tell me I have the red DNA, and identifying this would tell me I have the heterozygote. Imagine now repeating the assays for different type of these VNTR loci throughout the genome. So I may have a VNTR sample here, and another sample here, and another sample here, and so on and so forth. So at every point, then I'd be able to conclude whether do I have two red molecules or do I have blue molecules. Now remember, because I have a high degree of homozygosity, I don't expect to find the double band pattern too frequently like this one. But in the F2, this pattern would be much more frequent. So in conclusion, QTL can be mapped in a genetic population. It's a population that has different genotype. An example of that population is recombinant inbred lines. Recombinant inbred lines are advantageous. They are highly homozygous, which means that within a line, different individuals are nearly identical to each other, and they can serve as biological replicate in an experiment, giving me a high power to detect changes in phenotypic traits. Each genotype is then tested at the locus to evaluate whether that genotype, for example, little a versus big A is associated to a given trait value. If it is associated by a statistically determined difference, we can then conclude that we have a quantitative trait locus in that position. Under a QTL, we might have, and that is a lingo or a little bit of a slang term, under a QTL it means in the position on the chromosome defined by the QTL, we have one or more genes affecting the trait. So all this was enabled by the ability of genotyping.